Aravind from Welltech Multitech, Dr. Rangarajan, Dr. Sagundala Engineering College, Department of Electronics and Communication Engineering. We are here to present about cellular automaton based image segmentation algorithm for sickle cells. My batchmates are Balakumar and Rajesh. We are guided by Dr. P.G. Kuppusamy, Professor of ECE.
Our project major part goes in image processing techniques. Image processing techniques are widely used in the domain of medical science, detecting various diseases. The current system for the identification of blood parameters is costly and requires more time. The project deals with an image processing by Saga optimization technique and used for detecting abnormalities of blood cells in less time.
The main objective of our project is to diagnose the sickle cells in red blood cells, and to detect and cure a disease on time is very important in the field of medicine for protecting and saving human life, and to improve the oxygen carrying capacity in blood.
That's our optical coherence tomography capillaroscopy.
Blood consists of red blood cells, white blood cells and platelets. The damaged red blood cells are called sickle cell anemia. This sickle cell anemia causes many diseases and painful, and it reduces the life expectancy of the human beings and reduces the oxygen carrying capacity in blood. And finally it leads to cancer.
Process of separating or grouping an image into different parts. Image segmentation is also the process of dividing an image into multiple parts.
Cellular automata first proposed by one human. Why we are choosing this cellular automata means there are two principles. First principle, spatial structure. Second one, local interaction. Why we are choosing this cellular automata means after segmenting the images into pixels, the images look like a spatial structure. That's why we are choosing the cellular automata in this process. Cellular automata is a collection of colored cells on a grid of specified shapes that involves a number of discrete time steps according to the set of rules based on a state of neighboring cells. There are two states, on state or off state. Based on this state, we are processing our image segmentation.
First, we are considering actual urban pattern. Next, we are taking neighborhood effect. We are considering this both and we get simulated urban pattern. In this simulated urban pattern leads to Saga optimization. Then we are taking an actual urban pattern and difference between this actual and simulated urban pattern. Saga optimization, actual urban pattern and difference between the actual and simulated urban pattern has been taken into consideration. If it is minimized or if it is minimized, we are taking to optimal transition rule. Otherwise, it goes to transition rule, spatial constraints, topographic constraint and stochastic effect. Then we get the CA model configured using optimized transition rules. Steps involved in algorithm are image acquisition. We are getting an input image. Then we are reading the image. Reading the image means we are observing the image. Converting to black and white. Converting to black and white using grayscale. Image resizing. We are resizing the image. If we get 372 into 228, the pixels will not be equal. So we are getting 372 into 372. That is known as image resizing. Next, image segmentation. After segmenting, we can compare the pixel. Then we do masking and we get the output image. The algorithm are image processing toolbox, statistical toolbox, Dicon images, MATLAB software. Projector. Sickle cells can be detected in earlier stage. It is cost-effective technique detecting the abnormalities of the blood cells in less time. Main applications are it is used in the field of medicine. The output result is given into the laboratory pathologist to diagnose the sickle cell anemia.
For detecting the sickle cells based on cellular automaton, and the input image is, this is the input image. In this input image we have don't find the sickle cells and the damaged cells, but in the output image the sickle cells are clearly shown. The second output is epoch. In this epoch image the cells are overcoating. This is clearly shown. And the final output is mask image. In this image we have clearly view the output of the sickle cells. These are the sickle cells.
This video. Thanks.